So last fall, I became a first-time mom when my daughter Evie was born. And along with all of the joy and excitement that comes with new parenthood, there was also the natural concerns. What if I'm not doing this right? What if I'm never going to sleep again? But one of the things that I was not expecting to be worried about, but have been, is recurring infection. We have seen her pediatrician 10 times now since she's been born for the same very stubborn upper respiratory tract infection and chronic ear infection. And ironically, this hits very close to home because this is what I study as for my graduate dissertation, is recurring ear infections. So in our lab, we study non-typable Haemophilus influenza, or NTHI, which is a gram-negative commensal organism which asymptomatically colonizes the human nasopharynx. However, permissive risk factors can allow NTHI to migrate up the eustachian tube and into the sterile middle ear, where it can cause chronic and recurring otitis media. My project specifically focuses on understanding how NTHI pathogenesis changes in the face of nutritional immunity by the host. Nutritional immunity occurs when the host tightly sequesters essential nutrients that the bacteria might need in order to prevent microbial outgrowth in sterile areas. So NTHI, when it lives in the nasopharynx, it has access to the heme-iron that it needs to grow and survive. However, this bacteria cannot synthesize iron itself, and so it must get it from its host environment. And as it migrates up the eustachian tube and into the sterile middle ear, it becomes starved of heme-iron as a result of nutritional immunity. However, as otitis media progresses, increased inflammation will again provide a source of heme-iron for NTHI to grow and survive. And so this transient heme-iron restriction that occurs as NTHI migrates up to the middle ear is what we are interested in studying and understanding, because we've previously shown in our lab that this transient heme-iron restriction results in changes in pathogenesis, specifically characterized by changes in biofilm morphology and architecture, as well as changes in disease severity. And so in order to study this, we've developed an in vitro culture-based method in the lab where we culture the prototypical clinical strain of NTHI, 86028NP, which was isolated from a child with otitis media. And we culture this in defined iron source media in parallel cultures where one has heme-iron and one does not. And then after 24 hours, we transition both of these cultures back into media with heme. And this creates a continuously exposed and a transiently restricted culture with regards to heme-iron. And we can then use these cultures in downstream experiments to understand the pathogenesis of NTHI and how it changes as a result of this heme-iron restriction. And interestingly, when we use these cultures to infect our animal model of otitis media, which is a chinchilla, we see that it forms intracellular bacterial communities in the chinchilla middle ear upon transient heme-iron restriction. And this is really a striking finding. So in this image, you can see that NTHI is seen in green. All the arrows also indicate this. And the DNA is stained in blue, so you can see that inside the cell, as well as the cell membrane is seen in red. And I hope that you can see the multiple intracellular bacterial communities that are forming throughout this chinchilla middle ear mucosal tissue. And this is really a striking finding because Haemophilus has been classically considered to be an extracellular pathogen. It's not typically known to be able to survive intracellularly. Typically, if it's going to enter an epithelial cell, it will be quickly targeted for degradation. And so for us, this is one of the first times that we've been able to see possibility of NTHI being able to survive intracellularly. And this has big implications for kids that struggle with chronic and recurring otitis media, because if the bacteria is able to hide out in your cell during recurring courses of antibiotics, it might make the antibiotics less efficacious and might promote recurring infection. And so I'm interested in trying to understand how these intracellular bacterial communities form and how this relates to heme-iron restriction. And so in order to study this, we took a step back to look at the fate of this intracellular NTHI in epithelial cells in vitro. And so I hope that you can see on the left, these are cells that we have taken, these are normal human bronchial epithelial cell monolayers that we have infected with either our continuously exposed NTHI, so NTHI that's had heme-iron the entire time, or a transiently restricted NTHI, so this is NTHI that has been starved of heme-iron for 24 hours. And when we infect the epithelial cell monolayers with these cultures, we find that they have very different intracellular lifestyles. So on the left, you can see cells that were infected with continuously exposed NTHI appear to be contained within a vacuole or a membrane compartment. So you can see little clumps of bacteria within the cell here. And on the right, excuse me, when we infect with transiently restricted NTHI, we see the formation of these intracellular bacterial communities forming. We don't see that the bacteria appear to be contained within a vacuole or a membrane compartment. And so this suggests to us that these bacteria are trafficking differently within the cell. And so in order to look at that, we looked at the endolysosomal pathway. So NTHI it's been shown in the literature that NTHI can traffic through the endolysosomal pathway for degradation. So it will first reach the early endosome before reaching the late endosome, and then will eventually fuse with a lysosome, and this will result in degradation of NTHI and killing of the bacteria. And so we were interested in understanding if transiently restricted NTHI still traffic through this pathway in the same way, or if they might be able to escape from an early endosome or evade this pathway completely in order to form intracellular bacterial communities within the cell cytoplasm. So to investigate this, we took advantage of EEA1, which is an early endosomal antigen 1. It's a protein on the the surface of the endosome that we are able to use fluorescent markers for in order to look for colocalization of NTHI with this marker. So colocalization of NTHI with this marker would suggest that NTHI is able to reach this endosome. So we were looking for changes in colocalization between our continuously exposed and our transiently restricted NTHI. And so when we infect normal human bronchial epithelial cell monolayers with these two cultures, continuously exposed or transiently restricted, we do find that heme-iron restriction alters NTHI intracellular trafficking to the early endosomes. And so on the left, when we infect with our continuously exposed NTHI, we find that it does traffic to these early endosomes. In these images, NTHI is seen in green, EEA1 is in red, and the cell membrane this time is seen in blue. So any colocalization between NTHI and EEA1 will show up in yellow, and I hope that you can see that on the slides here, that we see a lot of colocalization of continuously exposed NTHI with EEA1. But on the right, when we infect cells with transiently restricted NTHI, we do not see colocalization of NTHI with EEA1, and this suggests that it is able to evade or escape from this early endosome as we expected, or hypothesized, excuse me. And so this naturally leads us to question if we can use this data to help prevent internalization of transiently restricted NTHI. So our next question then becomes, do these two bacteria traffic differently within, or enter differently within the cell in order to traffic differently? And so there are multiple mechanisms that the bacteria can use to enter the cell, and these are all seen here. And so our current experiments in the lab are working to systematically inhibit these different endocytic pathways in order to see if transiently restricted or continuously exposed NTHI traffic differently. And while I'm not showing that data here today, we do have promising data that suggests that transiently restricted NTHI might be able to use macropinocytosis in order to form intracellular bacterial communities within the cell, while the continuously exposed cannot. And that's an avenue that we are continually pursuing in the lab. And so why is this research important? Aside from the fact that I now have personal experience with this issue that many moms and parents and children struggle with, there are a couple reasons why I believe that we should continually be pursuing new and innovative ways to treat chronic and recurring otitis media. And the first has to do with antibiotic resistance. So you might not have a child that struggles with this, but I'm sure at some point in your life you've been prescribed an antibiotic. Otitis media is the most frequently diagnosed illness in children under the age of 15, and clinical management of this illness relies heavily on antibiotic use, often multiple courses, as is the case with my daughter. And so if we are able to develop a way or understand better how NTHI can hide out in these cells during antibiotic treatment, we might be able to prevent the internalization of bacteria into these cells, which will make current antibiotics more efficacious. We could also use the data that we are currently generating to help develop treatments that can target the bacteria to prevent it from internalizing at all. Secondly, chronic and recurring otitis media is really a quality of life issue. Many of these kids struggle with this for many years, and it can result in tympanostomy tube insertion, as well as hearing loss and speech and language developmental delays. And finally, this project has really changed the way that I view challenging the status quo. Many of the doctors that we have seen for my daughter's chronic ear infections say, you know, this is this is what kids get. They're going to get ear infections. It happens. She's in daycare. There's really nothing we can do about it. And my viewpoint on this has really changed to be, why is that the case? What if we can change that? And what if it doesn't have to be that way? And what if the research that is being done here at the Infectious Disease Institute can really challenge the status quo on chronic and recurring infections like otitis media? Thank you.